Hello, I am Carolyn Jerba. I am a senior director in the small molecule drug discovery group at BMS. And I'm very excited to be able to come and talk to you about some of the work that we're doing at Bristol Myers Squibb in terms of diversity and inclusion. And thank you to the organizers for organizing such an interesting event. So in BMS, what I figure I'll do is give you a quick overview of our company and then talk about some of our diversity and inclusion efforts. So within BMS, our mission is really to discover, develop, and deliver innovative medicines that help patients prevail over serious diseases. So we're really looking to find transformational medications and really looking to treat some of the most serious unmet medical needs. So within research and development, we work on a variety of different therapeutic areas, including oncology, which is our biggest therapeutic area. We also work in hematology, cardiovascular, immunology, fibrotic diseases, as well as neuroscience. So we also look at a variety of different capabilities to find drugs to treat diseases in these areas, including biologics, cell therapy, small molecules, as well as protein homeostasis. And protein homeostasis includes things such as protein degraders, PROTACs, and cell mods. So with this range of therapeutic areas that we focus on and capabilities, that leads to a really robust pipeline within BMS. So we have a number of leading products across 4 of our therapeutic areas, including Opdivo, which is one of our key oncology drugs. We also have a deep and broad late-stage pipeline, including 9 recent or potential near-term new product launches. And we're very excited this year to launch 3 new drugs, including Camzyos, Opdualag, as well as Sotyktu. I'm particularly excited about Sotyktu. This is our most recent launch, and this is a TYK2 inhibitor for the treatment of adults with moderate to severe plaque psoriasis. And this is a drug that was discovered by internal BMS scientists. We also have a robust early pipeline, including over 50 assets across phase 1 and phase 2 clinical trials across a variety of different platforms. So we really consider ourselves to be focused on patient-centric innovation. So really, again, trying to find the best medications to treat these indications in a variety of therapeutic areas, and really with the effort of trying to get these drugs to patients as quickly as we can. So within BMS, we're guided by a single vision, which is transforming patients' lives through science. And we do have a key goal of really driving equitable advancement and outcomes for all. So in order to do this efficiently, what we really need to do is focus on diversity and inclusion in all aspects of the work that we do within BMS. So in 2019, in building on a longstanding history of focus on diversity and inclusion, BMS decided to re-image and redevelop their global inclusion and diversity strategy. And so they were able to roll that out in 2020. So leading with our value of inclusion, our global inclusion and diversity strategy has 3 pillars focused on 3 critically important groups. And that includes patients and communities, really driving innovation and growth to address the unmet needs of our increasingly diverse patient population. We focus on our colleagues, making sure that our colleagues feel like they belong and they have a voice within the workplace. But also by having a diverse workforce, the employees within BMS can really speak to and advocate for the communities that we're trying to treat. And then also focus on business and industry, really trying to reimagine and transform our business and industry and business practices to make sure that we can achieve equitable outcomes and for all patients. So I'm going to spend most of my talk really talking about our efforts towards diversity and inclusion in clinical trials. But I do want to mention that there's a lot of other work that goes on within BMS around this area. So, for example, BMS has set aside $150 million to be spent over a period of 5 years in the form of grants, really to focus on these health disparity issues within the United States. Additionally, the BMS Foundation, which is an independent charitable organization, has also set aside funds, again, in terms of grants, in order to also address a lot of these issues, focusing on things including, sort of patient advocacy and education, increasing diversity in clinical trials, as well as clinical investigators, and focusing on also making sure that BMS has a very diverse global workforce. But as I said, the thing that I'd like to focus on is the need for diversity in clinical trials. So I'm actually a medicinal chemist, so I work somewhat indirectly in our clinical trials. My group helps deliver the drugs that are used in these trials, but this is an area I wanted to focus on because this is a part of BMS that I'm very proud of, and the work that we're doing in this area. So I think initially what I need to do here, though, is really describe and define what diversity means. I would say probably 20 years ago, we thought of diversity as being something more like gender diversity, racial diversity. But I think nowadays we really need to take a more expansive view of what diversity is, including looking at things like gender diversity, gender identity, disabilities, as well as socioeconomic status, to make sure that when we design our trials, we're really designing them for all patients and not just a subset of patients. And I think, as people have noted during the course of the global pandemic, it had been made really clear that there were some inequities in terms of access to medications and medical care treatment for certain communities within the United States. So this is something that we definitely need to address, and also need to address this because we've seen over time that America's racial and ethnic overall makeup has been changing over time. So in this case, I'm describing race as ascribed to individuals based on their physical traits, while ethnicity is often chosen by the individual. And ethnicity includes things such as, culture, religion, nationality, for example. And if you see the graph here on the right, this is from the Pew Charitable Trust, looking at the percentage of U.S. population based on race and ethnicity, starting from 1960 projected out to 2060. What you see is over the last 2 decades, there's definitely been an increase in the diversity within the United States, and this is projected to continuing to increase. And so this is something that we really do need to have a much larger focus on. So currently in clinical trials in the United States, patients are represented by about 80% white patients. And this is in contrast to the overall makeup in the United States. So for example, Black Americans represent about 12% of the US population. However, only about 4% participate in clinical trials. Hispanics represent about 19% in the US population, but only 3% participate in clinical trials. And Asians represent about 6% of the population, but only about 3% participate in clinical trials. So clearly there's some discrepancy here, which we need to try to address. So there's a number of reasons that could lead to this inequity here in clinical trial participation, and that's due to a variety of potential barriers to participation in clinical trials. So I'm not going to go through everything here, but I do want to highlight a few potential barriers. So in terms of the sponsors, there could really be a variable understanding of what the patient's needs are to participate and what effect it has on their impact to their daily life to participate in a clinical trial, as well as oftentimes lack of direct communication to the clinical trial sites about the importance of diversity and inclusion and measuring that metric during the course of the trial. In terms of the investigators, there's often lack of diversity in terms of both the investigators and the research staff. And because there is sometimes a lack of trust between certain communities and the medical community, this could lead to people not wanting to participate in the trial, or certainly lead to simply just unconscious bias and a lack of referrals of these types of patients into the clinical trials. In terms of patients and caregivers, there are a number of practical obstacles to participating in clinical trials. These include things such as transportation at the trial site, childcare while being treated, lack of insurance, and as I already mentioned, a lack of trust in the pharmaceutical and medical industry. There are also a number of potential societal factors. So qualified subjects aren't always offered the opportunity to participate. So sometimes the inclusion or exclusion criteria for trials automatically eliminate certain populations. And there are also a number of socioeconomic burdens to participating in a clinical trial. This includes things such as like the cost of transportation of the clinical trial site, the cost of childcare while being treated, as well as the potential cost of missing work while participating in a clinical trial. So there are many factors that potentially lead to lack of diversity in clinical trials, but BMS is really setting out to try to address some of these specifically. So BMS is committed to doing its part to help ensure that patients have a fair and just opportunity to achieve their optimal health outcomes. And so we're really working to try to improve the recruitment of a diverse patient population to make sure that the trial population is much more reflective of the real world population, which I've said has been changing over time, as well as the people affected by the diseases that we study. So we're really taking a foundational approach here. So this is a process-driven approach. So a lot of this is about really about change management. So working with the trial sponsors and investigators, having them sort of unlearn their typical ways of doing things and sort of relearn a more inclusive way to design the trials. These are really, this approach is aligned with our overall enterprise health equity goals that I mentioned, and it's really powered by our talent. So we do have people within BMS specifically dedicated to trying to address these issues. So the projected outcomes here are really to increase the opportunity for all patients, accelerate innovation and development. So for example, if we can treat a much broader patient population early on, we can very quickly determine if our drugs are both safe and efficacious in all patients and allow us to deliver our medications more quickly. And these outcomes would also be aligned with the health authority requests. So organizations such as the FDA are now requesting that we pay much more attention to the overall trial diversity. So for our progress here towards diversity in clinical trials, our main goal is to improve the diverse patient recruitment, again, ensuring that our participation, the trials, is more reflective of the real world population. And one of our key goals is that 25% of the US sites participating in new BMS clinical trials need to be located in racially and ethnically diverse areas. And by that, I mean areas with more than 30% non-white population. So we have a number of different tactics in order to try to address this, including the site and investigator selection, as I mentioned, trying to locate the sites in ethnically and racially diverse areas, but also making sure that we have investigators and researchers that are also ethnically diverse. We also want to take into account our protocol design considerations, really ensuring that our clinical trial protocols minimize some of the barriers that I talked about on the previous slide, and also follow the FDA's guidelines in terms of diversity and inclusion. We also focus on communication and really reaching out to the communities that we're trying to target, as well as partnering with external thought partners, community advocacy groups, to really make sure that we understand fully the needs of the patient population. There's efforts around patient support, including things such as providing transportation to a clinical trial, and also in cases, say, asking the clinical trial to stay open later so that the patients wouldn't have to miss work in order to participate in the trials. I mentioned we do a lot of training on diversity and inclusion and the importance of that, things like unconscious bias training. And then importantly, we really track our overall performance to really ensure that we maintain and reach our commitments. So with all that in mind, I just want to talk about one case study, kind of bringing all of this together. So I mentioned that we need to really understand the epidemiology of the diseases that we're studying. So, for example, there are a number of diseases that are more prevalent in either a gender or within a specific racial or ethnic group. And so we really need to understand that when designing clinical trials. So in this case, for example, it's been shown that prostate cancer is much higher prevalence in Blacks. And so more specifically, prostate cancer is about 60% more prevalent in Blacks and whites. And the reasons for that really remain unclear at this point. However, it is known that in currently Black patients participating in prostate cancer trials in the United States represent only about 4% of those being screened and only about 9% of those that are enrolled. So clearly there's a bit of a discrepancy there. So some of the tactics that BMS is taking to try to address this is really collaborating with advocacy and community-based organizations to create awareness about prostate cancer, as well as awareness about clinical trials. So doing things such as a prostate cancer and pancake breakfast at churches, as well as reaching out to local fraternities and barbershops to make sure that they're really reaching the right population. And then also making sure that the targeted sites are in areas with an increased Black population so that there's less barrier to actually getting to the trial site. So this has overall led to an increase in participation up to about 18% by Black patients. So we think this is a really good first step. This is now sort of our baseline and hope to continue to grow from there. So the other benefits, and besides increasing the trial participation, is really an increased interest overall in clinical trials among Black men through these advocacy efforts. And this also has allowed us to enhance our study designs, preparing for subsequent clinical trials, really based on the advice from these advocacy groups. So we really hope to continue to build on this model for the future, really consistently applying what we've learned for this trial and in other trials targeting other communities, really taking what we've learned here and applying those to those situations as well. So that's a sort of a summary of what we're doing in terms of clinical trial diversity. But another key initiative within BMS that I also would like to mention is our people and business resource groups, or otherwise known as our PBRGs. So these groups are made up of sort of volunteer employees, and they're really focusing on a single dimension of diversity. So people can belong to multiple organizations within the set. And these groups really offer several different benefits to BMS. So one is really advocating for and speaking for the internal workforce. So we do things like bring in outside speakers, do education around specific issues for these groups, as well as helping people find opportunities, based on through coaching and mentoring to really help people advance and overcome some of the barriers that these groups face. But another important part of what these groups do is really try to address some of the business-related issues and really help guide the overall business strategy. So, for example, the BOLD group, the Black Organization for Leadership and Development, this group really spent a lot of work working with the clinical trial teams in the last case study that I mentioned, on things of really making sure that we understand fully what the needs of the community are, but also down to the level of looking at the recruitment pamphlets and educational materials that we're putting out, making sure that they're culturally appropriate and that they will really speak to that specific community. One other quick thing that I'll mention is the Pride Alliance. This is a group that represents the LGBTQ plus community within BMS. This group has helped change some of our company policies, in particular making BMS take a wider view of what family means. The importance for this is that you can now allow other family members, more than we used to in the past, onto the employees' benefit programs. And so taking this more expansive view of what a family has been really helpful for our employees and their families. So these are a lot of really good groups. A lot of really good work is done within these groups, and I think has really helped BMS with our overall corporate policies and advocating for the diverse patient populations that we want to go after. And then just briefly, I want to talk a little bit about some of the efforts we're doing. A lot of these are much more local efforts around not only building a global workforce which reflects the diverse cultures, backgrounds, and experiences of our patients, but also retaining the set of key individuals. So we have a number of different ongoing events. One thing we did recently that was very cool was a virtual research and early development inclusive networking event. So this was a one-day event where we invited people in virtually to, we had talks about sort of what's going on generally in BMS, some of the types of research we're doing, but also gave people the opportunity to meet with various leaders within BMS across the different organizations, such as chemistry, biology, computational sciences. And it was a really good way to build networks. And these have already led to some continued conversations and potential mentoring opportunities. So that was a fun event that I hope we can continue. We also have a number of internship opportunities, really looking to target underrepresented students. So for example, in chemistry, we partner with the American Chemical Society on the One Bridge program, where we're partnering with UMass Amherst specifically to try to bring some of their underrepresented students into BMS for summer internships. We've also expanded that a little bit, and we've been able to donate some of our equipment to UMass Amherst to help overall support their chemistry programs. We also work with Project OnRamp, which is a local group that is looking to provide underrepresented students with paid internships over the summer in the biotech and pharmaceutical industry. And we did have 5 students in the summer, which was fantastic. So in addition to that, we do a lot of STEM outreach. That includes things such as an on-site science day, where we can bring in, for example, women in chemistry for a day of science within BMS, as well as focusing on STEM outreach to kids. So we've had some local kids on site and had a fun day of science demonstrations, but we also sponsor some events like the recent MIT Museum and Cambridge Science event, which is a huge event. We also have some employee grassroots D&I initiatives focused on inclusion, talent acquisition development, as well as supportive women and underrepresented ethnic groups. So this is a really fun group. They have a lot of different activities to engage employees as well as educate. And this group not only focuses on trying to increasing our diversity within BMS, but also making sure that we retain our diverse population. So that's a number of the different activities that we have going on within BMS in terms of diversity and inclusion. If you have more interest in any of these, I encourage you to visit our website at bms.com. If you go to the About Us page, you can actually find our 2021 Global Inclusion and Diversity Report, which really discusses all the efforts that we're working on and all the progress that we've made to date. So thank you.
Good afternoon, everyone. First of all, I want to thank MassBio for putting on this fabulous event and for the opportunity to take part in kicking off day one here on the oncology-focused day of Innovation Week as part of American Cancer Society. We couldn't be more pleased to be part of this event. I'm Steve Curtis. I'm the Director of New Ventures at BrightEdge, which is the impact venture capital arm of the American Cancer Society. And I'll spend just the next few minutes telling you a little bit about what we do at American Cancer Society broadly and BrightEdge more specifically. Bear with me while I get the slide share to work here. And hopefully that did it. Great. So, first off, let me give a little bit of the overview of the American Cancer Society. I think I can say this because our CEO, Dr. Karen Knutson, was actually just in Cambridge last week for an event that we put on and described it this way. This is not your grandmother's American Cancer Society. We've undergone through quite a bit of evolution over the past 100+ years that we've been in existence, but really over the last few, with a significant changeover of leadership and a real focus and commitment to some more progressive views on the ways that we can really impact patient care, their families, and across the whole continuum. So where do we exist? Well, we exist because the burden of cancer is unacceptably high. I think everyone that's on the line here today would agree with that. And our mission is really to improve the lives of cancer patients and their families. And we do that through 3 pillars. Our advocacy pillar through the Cancer Action Network, ACS CAN, through our discovery organization, as well as direct patient support. And at the bottom here, you see our vision statement, and it's really the 8 words that define us. Ending cancer as we know it for everyone. And the for everyone here is really highlighted because it is that commitment to equity and reducing disparities that really sets, I think, American Cancer Society apart. And it's something that we take very, very dear to heart when we are thinking about both how we work through our donations, through the philanthropic work that we do, but also from BrightEdge's perspective on the investments that we make. So let me describe each one of those pillars just a little bit more in depth and some of the work that we're doing to advance health equity and promote diversity. So on the discovery pillar, we have been funding some of the most important breakthroughs in both prevention, detection, as well as treatment and survivorship. Since 1991, we've put out over 13,000 grants and committed over $3 billion to extramural research. Actually, since the beginning that we've been doing this, it's been over $5 billion of the total commitment to grants. It makes us the largest nonprofit, non-governmental funder of cancer research in the United States. And as you can see here by these charts, a couple of things that we're quite proud of since that time in the early 90s, when a lot of important research has been coming out as to the causes and etiology of cancer, as well as amazing breakthroughs on the treatment and prevention. We've seen significant improvements in the cancer mortality rates. Now, we know that this isn't true for everybody, so I'll talk about that in a minute. By the numbers, here are a few that I've already mentioned. I won't belabor the point. We fund a lot of work. 50 of our Former grantees have gone on to win the Nobel Prize. ACS gets involved very early in the funding of these scientists, postdoctoral fellowships, research scholar grants, which are R01 type equivalents for faculty that are just starting their labs. Over 1,100 institutions and 25,000 researchers have benefited from ACS funding. But in order to be promoting the equity that we are looking for, both in health equity and delivering of cancer care, but also in thinking about research equity and providing the infrastructure and opportunities for everyone to get involved in this incredibly important work, we really think the intentionality here is important. So specifically in our extramural grants, we have created an entire center now dedicated to diversity and inclusion in cancer research. And we have just started to do a number of exciting new things, including these institutional development awards to HBCUs. We have also created a cancer health equity research center at a number of minority serving institutions. And in terms of the workforce development, we have created a research internship program and a number of new grants that will be announced shortly, specifically promoting equity, diversity, and inclusion in the way that we grant. And I think this is again one of these commitments and areas that we think is incredibly important as we think about really training the next generation of cancer researchers to make that next wave of breakthroughs. On the internal side, in addition to the grants that go out the door, we also have a team of internal researchers. One of the most highly cited researchers with an impact factor that is off the charts is Ahmadine Jamal. Dr. Jamal leads our cancer statistics and division. He is the one that probably many of you have cited anytime you've written a paper and looking for the latest incidence and mortality rates for a certain kind of cancer. This is something that we do on an annual basis and have been doing it for the last 50 plus years. Really breaking that down though is incredibly important. We have done some of the groundbreaking research to show the disparities in incidence rates and mortality rates across different ethnic populations, across different socioeconomic areas. It is really unfortunate that today, when we think about health equity at the American Cancer Society, we know that zip code, more so than genetic code, is one of the key determinants of whether or not you'll survive a cancer diagnosis. And that's just wholly unacceptable. So what are we looking to do about this? The other two pillars really are ways to directly impact patients and their families in the most immediate way that we can. So in addition to the research that we're funding outside the organization, as in addition to that, what we're doing inside the organization, our advocacy pillar, the Cancer Action Network, ACS CAN, is incredibly involved at every level of government, local, state, and of course the national government. We have presence in all 50 states and in all 3 branches. And some of the work that the advocacy pillar has done has really been to promote things like Medicare and Medicaid expansion, things that we know give the opportunity to promote and access to care for those that need it most. One of the other things, and our previous speaker from BMS did a great job highlighting this. Just taking one example, we have published on the racial and ethnic disparities among participants in precision oncology clinical studies. And this is something that now we know the FDA is looking for more diversity in those clinical trials. And this is important as we think about that next wave of breakthrough therapeutics, that they actually do benefit everyone who needs them. And this again goes back to our ending cancer as we know it for everyone. And some of the advocacy work that ACS CAN has done has been critical in making some of these policy changes. And then, because we know that research takes time, we know that changing policy takes time, we also have our patient support pillar. This is what provides sort of real-time support to those patients that can't wait. They have their diagnosis, the clock is ticking, they need our help. So we work in prevention and screening, lodging, transportation, navigation, survivorship, and education. Because we know that getting to your treatment, the ability to get to your treatment, to take that day off of work, to have someone who can drive you to that treatment is really important. So we have partnerships that provide what we call the Road to Recovery, literally volunteers driving cancer patients to their treatments that wouldn't otherwise be able to get there. We run over 30 Hope Lodges across the country. These are free lodging for patients that are undergoing cancer treatment. They're located near major cancer centers and provide that opportunity, again, for those that can't afford to stay near where they're getting treated and get the best care that they can. We are proud that we touch over 55 million lives per year and are active in 5,000 communities across the country. In addition, cancer.org, our website, as well as the Cancer Information Center, the number of unique visitors and calls that we get in on a daily basis are from those that have a cancer diagnosis and are looking to get that initial information. Where do they turn to? A lot of them turn to the American Cancer Society. I already mentioned the Hope Lodges, so I'll skip over this side, and the transportation with Road to Recovery. In 2022 alone, just this year, over nearly 25,000 patients have been transported to and from their cancer care through this activity. One of the things that I said, it's not just cancer patients, but part of our mission statement, cancer patients and their families, right? Caregiver support is also incredibly important. Whether or not someone has to take a day off of work to get to their treatment. So again, direct grants and other opportunities to support cancer caregivers is incredibly important and something that we are actively involved in. So the ACS equation is sort of summarized on this slide, right? We have our discovery pillar, putting research out the door. We have our advocacy pillar, molding and shaping policy so that we can provide more equitable care across the country. And we have our patient support pillar that is in real time supporting those gaps that we know exist. So, and that is really all adds up to our ability to work towards this goal of ending the 200 diseases that we call cancer as we know it today. So, with that, a brief introduction to BrightEdge. So, as I mentioned before, BrightEdge is the impact investment arm of the American Cancer Society. We invest in for-profit companies that are mission aligned with ACS and are really looking to fuel and accelerate ACS's mission through our investment and engagement with these companies. We know that actually moving the next therapeutic advance, the next screening advance to the clinic and to patients that actually need it is going to involve, a for-profit entity to actually make that happen. So at American Cancer Society, we're thinking about for-profit, nonprofit, and philanthropic partners that can, again, move this mission forward. So in early 2021, we sort of relaunched BrightEdge. The effort has been going on since 2019. We've made 18 investments. There's already been 3 IPOs from our portfolio, about $25 million invested. And we are really proud that we know that the products and companies that we're investing in are already impacting patients' lives. As we think about our investment portfolio, which is shown here, we are really investing across the entire cancer journey, from prevention and wellness to detection and early diagnosis. Of course, treatment. You can see a large portion of our portfolio is in novel therapeutics, but then also in recovery and management. Things like financial toxicities, right, and other issues that cancer patients face is incredibly important, and we're investing across that entire spectrum. What we're looking for here is what we define as really triple bottom line. So primary impact coming from the actual products and technologies and services that our companies are taking forward. When we look at a new investment opportunity, that impact lens is first and foremost. We want to make sure that what this company is doing is really going to impact cancer patients in an equitable fashion across the entire continuum. We're certainly looking for financial returns. And then the secondary impact of that is that those financial returns get reinvested into American Cancer Society so that we can do all of the great things that we talked about in those earlier slides across all three pillars. So by the numbers, what we've mentioned a number of these things before. One of the things that we are also building is that connectivity to our innovation ecosystem. Through the grantees and the other organizations that we fund, we are trying to bring forward the best and the brightest science, identify startups and companies that have ACS affiliations. We're mapping our grantees, working with them to understand where some new trends and evolving opportunities are, supporting those grantees that are looking to start companies. And we're building towards supplementing our impact venture fund with a new innovation acceleration fund that can really take forward the next wave of innovation that is coming out of the research that we already fund. This is another way to kind of make our donor dollars work even harder. So with that, I'm really excited to hear the rest of today's agenda. I thank you all for your participation and attention. To learn more about what we're doing at ACS and ACS Can, please visit cancer.org and fightcancer.org. Bright Edge Fund is at brightedgefund.org is the website for Bright Edge. It'll be undergoing some updates soon, so hopefully the new version will launch. And with any other questions, please feel free to email me directly. So thank you all for your attention.
That was a wonderful talk by Steve Curtis from ACS Bright Edge. Hello, everyone, and welcome to MassBio Innovation Week and Oncology Day at MassBio Innovation Week. My name is Gauri Nair, and I'm the Vice President of Innovation at MassBio. We are so happy to have the opportunity to connect with you today. Before we get on with our company presentations, I have a few housekeeping items. All attendees are currently muted to avoid background noise. For the startup pitches, each presentation will be 20 minutes, followed by a 5-minute Q&A. Please share your questions for these companies through the Q&A function on our Engages platform at the bottom of your screen. If you'd like to connect with the MassBio team beyond this event, please reach out to us at partnering@massbio.org. You can also find me online on the MassBio website. We are all there. Our emails are there, so please do reach out. With that, I'd like to welcome our first company for today, CellEvolve.
Good afternoon, everyone. Jarrell Porter, the founder, CEO of CellEvolve, and thanks for the opportunity to present a little bit on our company. I am going to share my slides. Just give me 1 second here. Again, thanks for the opportunity to tell you a little bit about CellEvolve. Definitely very excited to share a bit about our story and what we're trying to accomplish here. So first off, just a bit of background on how we see the opportunity, specifically in an area that we're pursuing and as it relates to oncology. First and foremost, I think everyone's well aware of the promise and the potential of cell therapy as evidenced by probably the most famous cell therapy treated patient in Emily Whitehead. However, there's a couple of unique characteristics of cell therapy and maybe appropriately for cell and gene therapy. Most of these programs are in smaller biotechs or academic institutions. And these are organizations that are not what we label as fully integrated entities, meaning that they tend to not have a commercial focus or commercial footprint. However, the industry is rapidly moving to a phase where that is necessary and needed. And that's what we label the fourth phase of the innovation cycle. And so this is where commercialization is crucial for these programs to become therapeutics. And so therefore, we founded CellEvolve a couple years ago to address this potential challenge, but what we see as an opportunity. So our founding mission is to accelerate the transformation of these innovations into treatments for neglected diseases. So we do this with a 3-part strategy. First and foremost, we work with the leading institutions in our space across the world, as well as the leading inventors and innovators. And you can see a snapshot of some of the institutions that we have partnered with to date and ones that we'd love to partner with in the future. Number one. The second thing that we do in the middle is we have a development and commercialization platform. And this is a platform that really allows us to take any cell therapy of any stripe, whether it's a virus-specific T cell, a CAR T, or an NK cell, and develop and commercialize that on the backbone that we're currently creating. And the most relevant part of that platform is something we call solution, which I'll talk about in a few minutes. And then last but not least, we focus on areas that we label as neglected diseases. And these are areas that for one reason or another, the industry has not pursued these areas, at least in the appropriate scale required. And there remains a pretty significant unmet medical need. And so that's what we label neglected diseases. Let me skip this one. So to date, we have signed 2 collaborations with 2 leading institutions. One is QIMR. The second one is an organization called Seattle Children's. And we're pretty excited about these partnerships because that has allowed us to assemble a portfolio of 7 programs with 5 of those in clinical development. Through QIMR, we've licensed a set of virus-specific T cells, one focused on JC virus, and the second one focused on BK virus. And the JC program is pursuing an indication called progressive multifocal leukoencephalopathy, which is a pretty rapidly progressive neurodegenerative disease, which I'll talk about in a moment. Our Seattle Children's partnership is different in the sense that it is targeting pediatric and ultimately adult brain cancers. And this is a very different cell therapy in the sense that it's a set of single targeted CARs targeting HER2, EGFR 806, B7H3. Also, there are a set of multiplex CARs, and multiplex CARs in the sense that they have 4 targets in one therapeutic. And it's the 3 that I previously mentioned, but the 4th would be IL-13R alpha 2. And this is a program that is preparing to enter the clinic as we just submitted the IND a few weeks ago, but I'll tell you more about that in a moment. So let me talk about our leading program. This is the Oncology Day. However, this is a cell therapy that's targeting a condition that unfortunately comes from a lot of the treatments for oncology, specifically heme malignancies, but also can just be related to the development of cancer in general. So PML is a rare disease, as I mentioned before. It's unfortunately progressive and largely fatal, and there are no approved treatments and no effective therapies for this condition. We have developed and are in the process of advancing an allogeneic cell therapy with a validated mechanism of action and a very, very compelling benefit risk profile, given that there's been a number of studies that use this mechanism to treat PML. We see it as a very attractive revenue opportunity with upwards of $2 billion in peak sales. And obviously we're talking about unmet medical needs, so sales is not necessarily the measure, the appropriate measure today. However, the point here is that there are a number of patients that are really in need of a novel therapy, and we see this as a very, very compelling opportunity. So much so that the FDA has granted us an IND clearance for our pivotal phase 2. As you all are aware, INDs are typically for first in human studies, but given the significant unmet medical need as well as the compelling benefit risk profile, the agency gave us green light to move forward with a randomized pivotal trial where we're preparing to start that study in the first half of next year. So as I alluded to before, and I'll focus on the pie on the right, unfortunately, PML is a condition that develops when there's immune suppression and typically chronic immune suppression. And unfortunately, as you can see in the blue, for heme malignancy, that represents a meaningful portion of patients that develop PML as a result of either the cancer itself or the therapeutics, unfortunately. And the challenge with this disease, as I mentioned before, is that unfortunately, most people pass away at a fairly short time period. So they die at a median of 4 months. And so, this is crucial for us to develop a new therapeutic to treat this condition. So I won't go through this in detail, but suffice it to say there is a number of studies that have confirmed the benefit of using this mechanism to treat PML. As you can see here, there are 6 published studies across a number of different academic settings, which gave us confidence to move forward, as well as the regulatory agency, as I mentioned a minute ago. This is our PML registrational trial that we call ASCEND-JC. I won't go through this in detail, but this is a global multi-center randomized trial, and it's randomized 2-to-1 with an adaptive design and has up to 60 subjects. And the intervention is a set of 4 infusions spaced out every 28 days. So you get 100 million cells per infusion that are HLA matched. And so, as I mentioned earlier, this is an allogeneic cell therapy. So therefore, it does not have all of the classic preconditioning or lymphodepletion requirements, as well as the post-administration monitoring requirements that you're all aware of with CAR Ts. Given that this is allogeneic, none of those are required. And therefore, it's a fairly benign administration of over 4 months with 100 million cells IV. The endpoint is time to progression as assessed by a scale called the Modified Rankin Score. And I won't go through any more detail there, but this is a validated measure that assesses neurological deterioration. So that's PML. But let me switch gears and talk about the second collaboration that we have, which is with Seattle Children's for what we call Brainchild. So Brainchild is a very novel program. As I alluded to before, it's a total of 5 different therapeutics that are all targeting brain cancer, initially focused on kids. And unfortunately, brain cancer is the top cause of cancer death among children. And there are several specific subtypes that are uniformly fatal. And therefore, this is a very, very important focus for us as a company. And as I mentioned, Brainchild is a very interesting program, five individual therapeutics. And ultimately, the goal is to develop this multiplex CAR that is going after 4 targets. It's delivered directly to the CNS and is delivered in an outpatient setting on a regular basis. We have 3 phase 1s that I mentioned before, and that's targeting B7H3, HER2, and EGFR 806. And ultimately, that's laying the foundation for the pursuit of this multiplex CAR. And ultimately, we think we have an opportunity to develop at least 2 therapeutics from this program, which I'll talk about in a moment. I'll skip this one. So the unique nature of this program is really designed to overcome some of the key challenges in CNS malignancy. First and foremost is tumor heterogeneity. So most cancers over time can express different targets, and that's very, very critical in brain cancer, and frankly has been one of the core challenges in developing an effective therapeutic. So we are going after the 4 most common targets in brain cancer, the 4 that I mentioned before. So that's number 1. The second challenge that we run into, and this is true for most solid tumor cell therapies, is T-cell exhaustion. And so our approach to addressing T-cell exhaustion is metronomic dosing. So we basically deliver a small amount of cells on a weekly or a biweekly basis until progression. And as you know, that's different than your traditional cell therapy, which is usually either one treatment or a limited number. This has a dosing schedule that resembles more traditional chemotherapy. The third challenge is the blood-brain barrier. So you have to obviously get the treatment to the area of interest. And the way this is handled here is we have intraventricular administration. So you have an Ommaya catheter or a Rickman catheter that's placed. And then after you manufacture the product, you treat the patient directly through the catheter in an outpatient setting. So as I mentioned, the three on the left that are in phase ones, and those are winding down. The fourth target, IL-13 R-alpha-2, was previously validated. So there was no need to repeat a phase one for that study. And as I mentioned, the goal is the development of this quad CAR, which is headed to phase one in March of 2023, as we just submitted the IND, and hopefully we'll hear feedback from the FDA shortly, ideally before the end of the year. So that's it on Brainchild. Let me tell you a little bit about our platform, which I mentioned before is called Solution. So platform is an opportunity for us to develop and commercialize, frankly, any type of cell therapy. And so this addresses obviously the core issue of manufacturing that you all know is definitely a mission critical area for cell therapy. And the way we've addressed that here at CellEvolve is we have a partnership with our academic collaborators, QIMR and Seattle Children's, who has their own manufacturing facility. But we also have a partnership with a newly launched manufacturing company called Resilience. So Resilience is our preferred partner for cell therapy manufacturing, and they are also an investor in CellEvolve and a board observer. And so we have a tight partnership with them to address all of our manufacturing needs. So that's number one. Number two is the technology. So you do need a technology solution to address many things in cell therapy, but most notably chain of custody and chain of identity. And so we've developed our own platform to address that need. And then last but not least are the people. And so our team will leverage all of these other parts of the platform and leverage the data that's generated through our system to develop and commercialize cell therapies over time. So as I mentioned before, we really lean on academic partners to develop and continue to innovate over time. You know, we've already signed 2 partnerships with QIMR and Seattle Children's, as I just described before, but there are others that we continue to pursue. And I only highlight this more as an illustrative set of examples, but we continue to look at areas that still have unmet medical need and where there's a meaningful amount of innovation. So for example, allogeneic CARs, I think we all know is an area that's very, very interesting and very important for the field. So we continue to look at that as an area to pursue. Although heme malignancies represent the initial focus for cell therapy with the CD19 and BCMA CARs, there are still a significant number of heme malignancies that have high unmet medical need and don't have cell therapies either being pursued or approved to date. Solid tumors obviously is the holy grail, as I mentioned with our Brainchild program, so we continue to pursue other areas there. And we're also looking at novel targets for some more competitive areas. Again, the key here is for us to focus on neglected diseases. Our team, I won't go through every individual here, but we have a cross-functional group of folks that have wonderful experience both in and out of cell therapy in both big and small organizations. Consistent with that theme is our board and our advisors. Our advisors are cross-functional. So we have very traditional scientific advisory board members like Dr. Bertania at Stanford or Dr. Irina Cortese at the NIH. But we also have CMC experts, given the criticality of CMC, and Patrick Hanley at Children's National, as well as Anshul Mangal at Project Pharma. And I already mentioned that Resilience is a board observer represented by Eitan Abraham. So our key milestones. So as I mentioned, we have the IND cleared from the FDA and therefore are preparing to start our PML pivotal trial. And we'll start that ideally in the first half of next year. And it's a relatively short study. As you can see listed here, we anticipate that we'll have a critical data readout in 2024, and that will allow us to move towards an FDA submission in the second half of 2024. And you can also see here the upcoming activity at the bottom for the Brainchild programs, both the IND being cleared for the Brainchild 4, but then also possibly pursuing Brainchild 3, which is the B7H3 targeted program for pediatrics. And so we're rapidly in the stage of preparing for those 3 clinical trials. So that's my final slide. Let me stop there. I will stop sharing.
So I will be asking you questions that are coming in through the live audience. The first question is, could you comment about the scalability of your approach?
The scalability, that was the question?
Yes. And this question comes from Harvard University.
Yes. So it's scalable, obviously, with sufficient capital is probably the gating constraint here. However, we're of the mind that we could probably handle a portfolio in the coming couple of years of anywhere between 5 to 7 clinical trials. And I think that the key here is staging those, trying to, if trials are ongoing, that's one thing, as opposed to trying to start multiple studies at the same time. That gets a little more complicated. And therefore, you would end up with a staggered potential commercialization schedule as well, which is the other key part on the opposite side of it. So, 5 to 7 is about the right number from our perspective in the near term. I mean, obviously, as we grow, that could grow as well.
I actually have a question. Can you tell me
I can't repeat that question for me, please.
So I actually have a question for you, which I'm going to ask while other questions come in from the audience. What does it mean for Cell Evolve to be on Innovation Week and to showcase your company through this program? We're so glad to have you here today.
Yeah. So first of all, thank you for the opportunity, and very excited to have a chance to present a bit about our company and what we're trying to do. And so, first of all, grateful for that. And I think it speaks to the wonderful meeting that you've set up, but then also hopefully is a nod to a slightly different approach for a company that you typically see in emerging biotechs and definitely in cell therapy. I mean, typically, there's usually a scientific idea and a thesis, and that is taken from labs or early discovery and move forward. We're kind of coming at it from a slightly different angle. And so I believe that that's part of the reason why, we were selected to participate. You wanted to hear a little bit about a different model. So we're grateful for that.
Absolutely. And you're really, your company is as a refreshing approach, and you're making a difference. In the
Absolutely.
past,
Thank you.
making a difference. So I don't see any more questions that have come in from the audience, but I'm sure that there will be some. If anyone wants to reach out to Salabol, please reach out to the MassBio team, and we'd be happy to connect you if you have any follow-up questions, and I'm sure they'd be happy to answer. Moving to our next presentation.
Thank you. Thanks for the opportunity.
Yeah. Thank you. Thank you, Derek. Our next presentation will be a company, Glycopath, and I'm very happy to welcome Anand Mehta from Glycopath, who will be our next presenter. Thank you.
I'd like to start by thanking the organizer for allowing us to present today. So my name is Anand Mehta. I'm actually a professor as well in the Department of Cell and Molecular Pharmacology down at the Medical University of South Carolina. And I'm one of the co-founders, along with two other faculty members here, of that has a very unique technology platform that targets a certain type of molecules that have been, I'll say, not ignored, but not paid that much attention to, but are now actually becoming quite a hot topic thanks to a recent Nobel Prize winner out of Stanford. So what I'd like to talk to you today about is really, one, the actual molecules we're looking at, and then introduce the actual platform that we developed to allow us to look at these molecules, and then talk about how we use this technology specifically for the early detection of liver disease. And liver disease is something that I'm very interested in. This is a platform technology we're going to use first for the early detection of both liver fibrosis, liver cirrhosis, as well as liver cancer. And this, again, I'd like to point out, is not just a one-use technology, but really becomes a technology platform that I think that can be used for a lot of different disease states. So, liver disease is one of the largest rising diseases worldwide. And historically, it's been driven by viruses. It's been driven by hepatitis B virus. It's been driven by hepatitis C virus. And those two are becoming less and less in the US, and they're becoming less and less worldwide. And that's really down to the effective development of a vaccine for hepatitis B virus and for effective treatments for hepatitis C. However, even though those two etiologies of hepatitis are being taken care of, liver disease isn't going away. And it's not going away because it's being replaced by the Western diet. So the Western diet has historically been thought of as a high-fat, high-sugar diet. It's a little bit more complicated than that. It's primarily a high-fat, high-sugar diet with a moderate amount of use of alcohol. So I'm in Charleston, South Carolina, and that's our diet. A very high-fat, high-sugar diet with, an occasional drink along the side. And liver disease is progressive, and I'll show you some of the data for that in a second. It's not something that just happens like that. Even viral hepatitis is not something that happens like that. It takes a long period of time for these diseases to start manifesting themselves and causing problems. And in all these cases, if you've been diagnosed with liver disease or at a risk of developing liver disease, you need constant surveillance. And that's where a lot of our technologies come in. So even with the effective vaccine, there are still about 2.5 million people in the US that have chronic hepatitis B. All of those people are at risk of developing end-stage liver disease in the form of liver fibrosis or liver cirrhosis or liver cancer to a certain extent. And they have to be surveyed every 6 months for the development of these progressive diseases in some ways. There are almost 3.2 million people in the US that have chronic hepatitis C who have been potentially treated for their hepatitis C and cured of hepatitis C, but their underlying liver disease still exist, and they will still have to be surveyed every 6 months to every 2 years for the monitoring of their liver disease, whether it's fibrosis, cirrhosis, or cancer. And so they have to be screened constantly for that. There are now over 100 million people in the US who have what's defined as fatty liver disease. That's either non-alcoholic steatohepatitis or non-alcoholic fatty liver disease. And the rate of fibrosis, cirrhosis, and cancer is lower in those patients, but the sheer number of those patients are going to be a huge economic burden and a huge medical burden in the US in the coming years. And so there is right now a huge number of patients that need to be surveyed for the progression of liver disease, as well as a huge number of people that are coming in the future that will swamp the existing methodologies that are used and require new things and new ideas to effectively monitor them. Our healthcare system is in no way, shape, or form ready to deal with 100 million people coming forth that have to be monitored for liver disease. That's just not the way it's set up right now. The one last thing I'll point out, actually shown here in the bottom, again, this is progressive liver disease. So you do not go from having a hamburger with a beer to developing, cirrhosis. It's a progressive thing that takes many, many years to occur, where you start with fatty liver, and a period of 10, 15, 20 years, your liver will get progressively more fibrotic. So you'll have scar tissue that starts building up in your liver, where eventually the majority of the liver is comprised of nothing but scar tissue. That's cirrhosis. And when you have cirrhosis, your rate of developing a liver cancer is about 5% per year. And at that point, cirrhosis and/or hepatocellular carcinoma can both kill you, and that's the big problem. And currently, the recommendation is if you have cirrhosis, you should be screened every 6 months for development of liver cancer. And if you've got fatty liver disease, every year or so, you should be screened for the development of progressive liver disease in terms of fatty liver disease going to fibrosis or advanced fibrosis, eventually cirrhosis. So there's a huge clinical opportunity to do something and intervene to then manage the patient, but there's also a huge commercial opportunity to develop develop surveillance methods that can be applied to this patient population. And like most things, early detection is essential, and it's really essential in terms of liver cancer as well. And that's best shown by this graph that's been published by a number of people, including us, where if you have a small liver tumor, for example, and that's detected early on, where it's less than 2 or 3 centimeters, you can cut out that tumor, and that patient basically can survive for another 10, 15 years, 20 years plus, and continually be considered basically cured. In contrast, when that tumor is caught late, survival is very poor. And so early detection is absolutely essential for this patient population. And so that's why all major societies that manage these patients recommend active surveillance. So surveillance every 6 months for the development of liver cancer, and at least every year for the progression of liver fibrosis. And so really, these methods and biomarkers that can measure these methods are greatly desired. And so there is right now still currently a great desire to have biomarkers that can identify those that are at risk of developing HCC, that is those patients that are getting progressive fibrosis, and then 2, methods that can detect early HCC so they can actually surgically resect it and actually improve patient outcome. And the approach we take is a little different, and it's this thing that we don't call it, but it's known as end-link glycosylation. And glycobiology and glycosylation is I think Carolyn Bertozzi just mentioned, is a sort of the dark matter of the protein world. And what it is basically just sugars, oligosaccharides, that are attached in a very specific way to proteins, creating what are known as glycoproteins. And it's been known for years that these sugars on proteins can change with various diseases. So many of you may have heard of these sort of glycan changes and never really realized it. CA19-9, which is a common biomarker for many cancers, pancreatic cancer, cholangiocarcinoma, other things, is nothing more than a carbohydrate antigen that's measured on a lipid or on a protein. These things have been known to change in many cancers, and there are literally tens of thousands of publications describing these changes. In some cases, they've been translated to the clinic and are used clinically, but in most cases, they're not. And the reason behind that is quite complicated. And it really comes down to the fact that it's not trivial to analyze these sugars. And so generally what's been done is that when people have done sugar analysis on a given protein, to act as a biomarker for something, they would have to purify out that protein through a patient sample. So in large quantities, you would have to then release that sugar molecule from that protein and analyze it through some methods. And historically, this was something we would give to a PhD student or somebody like that just to have them do. It's not something that you would do routinely on a high number of samples and multiple different proteins. It was not technically feasible to do. And that has really limited the field in many ways to sort of translate these findings that almost all academic labs have, literally tens of thousands of papers describing this that have had. People have decided to use other methods. There are things that we call lectins. These are sugar binding proteins that people have used in assays that can give you an idea of the sugars that change. And to a certain extent, they are okay. But again, they're not that specific. And the lectins themselves are not commercially grade reagents. They are really just purified from mushrooms and other things. They're not really suitable in many ways for a modern clinical diagnostic laboratory. And so it's not something that has gained wide use. And these approaches really are never really going to go that far in many ways. And they haven't for over the years. So we've developed a method that we think can and sort of addresses a lot of these limitations. So here at the Medical University of South Carolina, myself, along with who are professors also here, and who are world leaders in the area of spatial omics and spatial mass spectrometry, method that really leverages existing mass spectrometry workflows that are designed for tissue imaging, but utilize them and modify them to create a new proprietary liquid biopsy assay that we think has great clinical value. And it really just takes an antibody array that has antibodies to any given protein that you want. And these are, in some ways, commercially available. There are commercial sources that will actually print these arrays for you. They can be done on a nanoscale, on a picoscale, that make it quite easy to do and very easy to have lots of replicates and a lot of QC standards. But basically, you take an antibody array like this, you add protein, and then the key step here is we add in a special enzyme called PNGSF prime, which is an enzyme we made in-house that works on this glass slide to remove the sugar off of the antibody captured protein. That glycan that removes sugar stays exactly where it was removed from. So if it was removed from this spot in the top left-hand corner, it stays in that exact spot. If it was spot in the bottom right-hand corner, it stays in that spot. So you can localize a glycan removed from an antibody capture protein quite easily, and using a standard MALDI mass spectrometry imaging workflow, identify the sugars that are attached and get a diagnostic biomarker out of it. So I'll show you an example of what this looks like in a very simple way. And I'm showing just two proteins here because it's just easier to kind of get the idea of what this looks like with two proteins. So on the left here in panel A, you see like a glass slide with little chambers. And in these chambers, we've spotted antibody to two proteins, to a protein called alpha-1 antitrypsin, which is a classic liver protein, and an antibody to human IgG. And I'll get into why IgG in a second. And so you can spot those antibodies in distinct areas, and then you can image the sugars that are attached to them using the workflow I just described. And these two antibodies are next to each other, but for example, you can clearly show that if you look at panels B and C, the sugars that are well known to only be associated with one of those proteins, with alpha-1 antitrypsin, only shows up in the spot where you have antibodies to alpha-1 antitrypsin. That's shown in panel B. And in panel C, the antibodies that capture IgG have glycans that are only associated with IgG. And so you get very clean capture of specific proteins, and then you get glycan analysis that's associated with that captured protein. And so you can do that with one protein, you can do that with two captured proteins, you can do that with thousands of captured proteins. So whatever you want to put on that slide, whatever antigen you want to capture through an antibody, you can capture it and get analysis on the sugar attached to that protein. And you can do that for one sample, two sample, 100 samples, how many other samples you want in a high throughput way. Now, the great thing about this is that you can also leverage existing data. So our finding, for example, is our development of this assay is really a method. It's a method that overcomes a challenge that's existed in the field of how do you do glycan analysis on a large number of samples using a large number of proteins at once. That's something that was not capable of being done, but we can do it. And we can leverage other people's data. So for example, it's been published by our group as well as many other groups that the sugars on human immunoglobulin change with liver fibrosis and liver cirrhosis. And there are literally tens of thousands of samples that have been analyzed that have shown this. But it's not been clinically translated simply because it's a royal pain to do, and it's never going to work in a clinical setting. However, our assay now allows that to be done. So we can develop systems where we can measure these changes on specific IgGs, and I'm not going to go into too much detail about this, but we can see changes on people with healthy livers, that people that have early fibrosis called stage 1 and 2, late fibrosis, stage 3 and 4, where you see changes in specific sugar structures in IgG. In this case, we can look at IgG, IgG1, IgG2, IgG3, IgG4, IgM, IgA, etc. And we can see specific changes that occur on all of these proteins with the development of liver fibrosis and liver cirrhosis. And again, we're talking large data here. So you're talking, in this case, 5 proteins with about 20 glycan changes. So anywhere from 5 to the 20th power of combination. And from that, you can make a nice diagnostic algorithm where you can detect people with early to moderate fibrosis to those that have no fibrosis with very high sensitivity and specificity values, where you can detect patients with more significant fibrosis from mild fibrosis with very high sensitivity and specificity values, and detecting those that have very late fibrosis cirrhosis from those that have none with high significance and sensitivity and specificity values. Again, in many ways, from a diagnostic biomarker idea, we're not developing anything rapidly new. We're just enabling a lot of the technology that has never gone anywhere because it could never really be done in a practical way. We can now do that, and we can get data like this that shows fantastic biomarker potential. We can move beyond things like IgG and look at the literature. Most of this stuff is published. For A1AT, people have published that these changes occur with hepatocellular carcinoma. A1AT, there's over 1,000 patients that have been examined. With haptoglobin, again, another 500 patients that have been examined by various groups, independent of us. Hemopexin, over 1,200 patients. So again, there's a wealth of literature out there that's already identified these changes. They just have not been able to translate that into an assay that can be used in the clinic because the technology wasn't there. We now have that technology. And so as a proof of concept, we did another study where we looked at roughly 140 HCC patients, about 270 cirrhotic patients. These ranged in etiology from viral hepatitis to alcohol to fatty liver disease. And by looking at all these proteins in combination, we see changes on many of these proteins. Many of the changes are, I wouldn't say expected, but they're what have been reported in the literature, and we see those same things. And we can get diagnostic algorithms that are substantially better than what's clinically used now. In this case, AFP, alpha-fetoprotein, is the clinically available diagnostic that's used right now in the clinic, and we are a magnitude order better than that, and we're actually getting even better than this, simply because we're not just limited to now 5 or 6 proteins. We can literally expand this to as many proteins as we'd like. So again, I like to point out, this is a core platform technology. We're doing this here now for liver disease, and that's our first goal. We're doing that because we can leverage the existing data that's there in the literature that we've done, others have done, and apply our technology to bring that to market quickly. And again, I would also point out that there's a huge market there just because the numbers are large in the US, and also the clinical recommendation for surveillance is already there, and there is an unmet need for developing non-invasive markers for that surveillance, and we believe we can enter in that platform. So in conclusion, I would say that these assays are showing great promise. We're finalizing some of these before doing our clinical, pivotal clinical studies. One of the goals we have is to achieve slides that are prepped by a commercial vendor that come basically prepared, that are just basically take out of the kit and apply. So we're breaking these down to kit-based systems. These work on Bruker mass spectrometers that are now clinically available and that are becoming more and more widespread. And so this is not something that is pie in the sky thinking using instrumentation that is not really feasible. These are now out there and they're used. And so, and again, I'll point out that glycan alterations like this have been seen in many cancers. And so whether it's liver cancer, whether it's breast cancer, whether it's prostate cancer, whether it's colorectal cancer, whether it's Lyme disease or rheumatoid arthritis, this same core technology can be used in those same areas to be used to develop a biomarker of those specific diseases. It's the platform that's unique here, not necessarily the outcome, and not necessarily the diagnostic marker, which is itself can be unique too. But it's the platform technology that we have that I think was the greatest value. So in our eventual workflow, we are taking a CLIA path initially, where we'll get orders and run these assays in a CLIA lab that we'll set up. And the hope is that this glycopath disease score will be used to help the management of patients that have liver disease and guide that physician to the best and most appropriate treatment for that individual and that patient. And that's the goal that we're trying to do. So with that, I will end, and I apologize for running a little bit over time. Hopefully you were able to see that and hear that. And if you have any questions, I would be happy to take them now.
Hey everyone, Anand, that was a great presentation. Just waiting for questions to come up from the audience. I encourage the audience to put questions in the chat. Looks like we actually don't have any questions that came in, so we are just going to move on to our next presentation. And I'm very pleased to introduce NanoCan. And we'll soon have Eric Broyles and Adam Schilling from NanoCan joining us.
Hello, my name is Eric Broyles, and I am the CEO and founder of NanoCan Therapeutics, which is a company that Harvard's Dana-Farber Cancer Institute has spun out a patented technology that we believe will have a revolutionary impact on cancer care. In the 1990s, when my own father was diagnosed with lung cancer, the one thing that haunts me most about that horrific ordeal was hearing his doctors utter the words, "There's nothing we can do." I wake up every single day laser focused on advancing NanoCan's novel drug delivery platform so that no patient or family member ever has to hear those words again. The introduction of immunotherapy over 30 years ago was the biggest breakthrough in cancer care at the time. Blockbuster drugs like Keytruda and Opdivo generated in excess of $25 billion in revenue last year alone. But the problem with these potentially life-saving drugs is that they're given systemically through an IV, and then they bombard the whole body with repetitive high drug payloads. And in many instances, the drugs don't even end up where they're needed in the body to properly treat the cancer. And because of these toxicity issues that are often generated with current immunotherapies, the vast majority of cancer patients, about 80% of cancer patients, cannot avail themselves or realize the benefits of immunotherapy until now. Using our nanoengineering, our world-renowned scientific partners at Harvard's Dana-Farber and Johns Hopkins have developed and tested NEST, our nanoparticle encapsulated smart technology. This is a patented and proprietary delivery system that overcomes these toxicity challenges in order to finally deliver on the promise of immunotherapy. At nanoscale, NEST delivers sustained microdoses, only about 1/6 of immunotherapy that you compare to an IV, but it's directly where it's needed at the tumor site. NEST is a drug delivery mechanism that actually can be loaded with many different therapeutic agents. It's actually agnostic in terms of that. It becomes a drug once you load the agent in, it's considered a drug. But the company has actually had interest from many other pharma companies for the delivery platform for non-cancer indications as well. NEST has delivered some amazing preclinical animal results. Essentially, this product has been able to eradicate cancer cells at both the local site where it's treated and metastasize spread. And as you can see, the lifespan of treated subjects with NanoCan's technology far exceeds what's being delivered with current therapies on the market. NanoCan has the global exclusive license of this patented delivery platform from Harvard's Dana-Farber. More than $7 million of NIH and grant funding has been invested into the product, the development while it was still in an academic setting. And NanoCan was recently awarded $3 million in NIH funding to actually advance this product to clinical trials. The product covers 7 solid cancer types, with an addressable market of over $40 billion. And we maintain strategic, sponsored research agreements with both Harvard and Johns Hopkins for continued product development. NanoCan has a world-class management team that includes top scientists. So Dr. Paul Nguyen, who many people consider one of the top prostate cancer doctors in the United States, is a medical advisor to the company. Dr. Adam Schilling, who's joined me here today, has done over 10 cancer IND and clinical trials, and including registration of drugs as well. We have a world-class scientific advisory board as well, coming from top institutions, including MD Anderson, obviously Harvard, Memorial Sloan Kettering, and Cleveland Clinic. These advisors are very active and involved in the company and have been very helpful to NanoCan's advancement to where it is currently. NanoCan, our current stage is that we've actually accomplished a lot since we founded the company in 2020. We actually have already held our pre-IND meeting with the FDA about a year and a half ago. We have actually begun the GMP manufacturing of this NEST product. And we've, again, been awarded multiple NIH grants. And we're ready to begin phase 1 clinical trials early next year. And we're going to tackle hormone-resistant prostate cancer for our phase 1 clinical trial. we're also actually closing out a seed round. We're raising $3 million at a pre-money valuation of $22 million. And this funding, once we close this seed round, when we include that capital with the $3 million from NIH and current funds from current investors, we'll be able to actually go through and complete our entire phase 1 clinical trial. Obviously, we'll do the manufacturing and run the trial and complete the trial. So in summary, our novel patented NEST delivery technology has the potential to change the dynamics in oncology and finally deliver on the promise of immunotherapy. Thank you for the opportunity to share information on our company today, and I look forward to answering any questions in the breakout room.
Hi, Eric. That was a great presentation. Thank you.
Thank you.
I'm waiting for questions to come in, but in the meantime, please let us know. So what does Innovation Week mean to you? We have an underlying theme of diversity drives innovation, and we're so glad to have you here. What does it mean to be part of this and showcase your company's platform?
Well, I'm honored to be a part of this. Obviously, I have great respect for your organization, and it's an opportunity to just share what we're doing and obviously hear about other great innovations on the market or in the market and coming to the market. So it's always, again, a pleasure to be able to share with peer companies that are sort of going through similar challenges, and as we try to roll out our products to the market.
So I don't think we have any questions coming in. So we'll take a short pause and then we'll bring our next company in, which is Gurkhan Therapeutics.
Hi there.
Hi, Mohammed.
Great.
I'm going to leave this and I'll leave you to start your presentation. Thank you for being here.
Thanks very much. First off, thanks very much for giving me the opportunity to present our work today to such a great audience. So without any further ado, just go ahead with sharing screen and talking a little bit about Gurkhan and our technologies. There we go. Perfect. Just going on a laser view here. Perfect. So just do a little bit about the Gurkhan Therapeutics. It's a University of Toronto spin-off biotech company. We have R&D sites at Lab Central at Cambridge, Massachusetts, as well as at JLabs Toronto. And our mission at Gurkhan Therapeutics is to solve the problem of tissue and CNS selective drug and gene delivery. As you know, lack of efficient delivery of therapeutics is still one of the major challenges in clinic. Very limited amount of the injected therapeutic doses gets to the target tissue, and less than even negligible amount, less than 0.2% of the biological therapeutics reaches the CNS area. To solve this issue, as you know well, nanoparticle technologies been proposed to improve the biodistribution and the release rate or sustainable release rate of the small molecules and biologicals. And LNPs have been, again, more successful and made more traction in the clinic, mainly due to their great reproducibility and scalability compared to polymeric-based nanoparticle technology. However, the high immunogenicity and liver accumulation and toxicity is still one of the major challenges using these platforms in the clinic, particularly on the gene delivery side. At Gurkhan Therapeutics, our scientists have developed the next generation nanoparticle structures, which is a hybrid polymer lipid nanoparticle systems that facilitates extrahepatic and site-specific or tissue-specific delivery of payloads with an excellent safety profile, which has been validated in a repeated dose manner in GLP settings. Speaking of the key features, as you can imagine, this technology is extremely biodegradable, biocompatible, but more importantly, it facilitates active transportation across the blood-brain barrier. And it improves the delivery of the payload more than 50-fold higher than when you inject it free. It carries multiple targeting molecules on their surface that allows conjugation of targeting molecule of interest for more tissue and cell-specific delivery. And imagine all these unique features combined with its scalable manufacturing method really position TURP to develop a pipeline of best-in-class and first-in-class therapeutics for a wide range of therapeutic applications. The core mechanism of blood-brain barrier transportation really relies on the absorption of this nanoparticle, the absorption of ApoE proteins during the circulation on the surface of the TURP system that facilitates the recognition by the LDLR or LDL receptors and active transportation to follow an active transportation inside the brain. So it's technically ApoE LDL receptor. LDL receptor transcytosis mechanism. What is really cool about this mechanism is that we are recruiting endogenous source of ApoE rather than conjugating ApoE and then process the administration. That allows to mitigate any potential risk of toxicity where you see with transferrin or insulin-based compounds, and also enables exercising the full capacity of ligand receptors on the BBB endothelial systems and not competing against the existing proteins in the body. We also can get advantage of upregulated expression of ApoE. As you know, there are lots of reports that shows that the ApoE increases by 150-fold higher in patients with CNS disorders. So we can get advantage of that and accelerate the penetration of our payloads using the TURP system. This nanoparticle structure has been highly de-risked, advanced to the post-GLP GMP setting, and we've been used to deliver a wide range of payloads from small molecules to antibody and genes, RNA, mRNA, and siRNAs. Our strategy at CureCan is really to maximize the value creation by developing a pipeline of TURP-enabled products and unlock the full capacity and unlock the full potential of our TURP platform technology. And we have a hybrid business strategy. One is, co-development, out-licensing strategy with partners to developing novel TURP-enabled siRNA and mRNAs for multiple in a wide range of indications, as well as in-house product development. We have 2 products in pipeline. One of them is a cancer chemotherapeutic agent, TMX. That's a first-in-class oncology therapeutic anti-hypoxia immunomodulator that can turn cold tumors hot. The first indication we're pursuing that I'm going to talk more about that is a combo therapy with radiation therapy. And as soon as we collect this, the preliminary efficacy signal after finishing off the phase 1b in combo with radiation therapy, we'll explore the combo therapy of TMX in combo with immune checkpoint inhibitor, which has a massive market in the immuno-oncology market. The second product is TTRA, TURP-enabled trastuzumab, a best-in-class trastuzumab formulation that can crosses the BBB and target and treat brain metastases. TURP has shown great promises as a potent and safe platform for delivery of the genes. Unlike LNPs, as you know, that the phospholipid is exposed during the circulation after the administration. In the TURP system, we have this novel surface chemistry, which is negatively charged, sitting around the core, around the rim of the nanoparticles, and holding this phospholipid and the RNA payload at the core. So the TURP system sitting around and encapsulating the phospholipid. That enables us to first have extrahepatic delivery with a negative charge, and not having the lipid portion exposed. And also at the final step during the manufacturing method, you can lyophilize this system and improve the stability and the shelf life of the final product. We have done a series of in vitro and in vivo work developing TERP-enabled siRNA and mRNA, showing great successes in silencing the target gene and expressing the protein of interest using the mRNA in vivo. And also we've been recently transferred siRNA successfully to the CNS area and silenced the target gene in the brain tumor. So we're pursuing, as one of our strategy here at PureCan, co-development opportunities with biotech companies to develop novel TERP-enabled mRNA agents for a wide range of indications. We're also working with cell manufacturing companies to develop in vitro and in vivo transient CAR-T or CAR-NK cells to improve the safety and also cell killing efficacy of these products. I'm just going to be a little bit more focusing on our lead asset, which is TMX, is designed to overcome treatment resistance caused by hypoxia. As you know, still one of the major challenges we have in clinic is treatment resistance, and the root cause of treatment resistance is lack of oxygen inside the tumor environment, known as tumor hypoxia. Under a hypoxic condition, cancer cells produce byproducts such as hydrogen peroxide and change their gene expression patterns that promotes proliferation, immunosuppression, and angiogenesis. In cancer therapy, we're really good at drugging the top of this oncogenic funnel, and by that I mean targeting the receptors on the surface of the cells, or those in the cytoplasm, or those signal transduction targets. But we've not been able to target hypoxia, the key driver of treatment resistance in tumors. There's been a strategy to overcome hypoxia indirectly using anti-angiogenic drugs or chemotherapeutics such as hypoxia-activated prodrugs or HIF modifiers. But as you know, there have not been a great success in improving the tumor oxygenation, and most of these strategies been toxic at the efficacious dose or after repeated dose administration. So still the challenges exist there, and we're looking for the best solution. And here it is, TMX, our first-in-class, it's really positioned as a first-in-class IV injectable compound that can turn cold tumors hot by oxygenating the tumors. This agent is composed of TERP as the carrier system, loading manganese dioxide as the active ingredient. And the use of these active ingredient, manganese dioxide, was first introduced by our CSO at the University of Toronto. And since then, it was followed by hundreds of institutions, the use of manganese dioxide to practically oxygenate the tumor and eradicate hypoxia. So after administration, these agents gets accumulated inside the tumor, reacts with cancer cells byproduct hydrogen peroxide, and generate oxygen locally inside the tumor and scavenge hydrogen peroxide, which is a ROS, or reactive oxygen species. By eliminating this hypoxia-induced treatment resistance or immunosuppression, or let's say, put it this way, by simply oxygenating the tumor and scavenging reactive oxygen species, we can further damage causing lethal damages to the DNA of the cancer cells in combo with radiation therapy, and also changing the population of macrophages inside the tumor and activate immune cells, CD8 plus T cells, which all leads into activation of innate and adaptive anti-tumor immunity. And because it's a manganese-based agent, it also can be imaged, facilitating real-time target engagement determination. It also shown great promises, preclinically empowering the efficacy of radiation and immunotherapy PD-1 inhibitors by 5-fold, and also achieving complete tumor eradication compared to conventional standard of care combo therapies using anti-VEGF plus radiation therapy, which is reported to only have 2-fold improvement in life expectancy. So simply, these agents offers a series of advantages over the current products in pipeline or in the market. First off, it can work for a wide range of solid tumors and can activate anti-tumor immune response simultaneously. It also facilitates image-guided radiation therapy or an allowing image-guided and precision medicine strategy. The other interesting feature is that it also creates a unique business development opportunity for CureCan because we can pursue the application of this agent in combo with immune checkpoint inhibitor, where there is a huge unmet need. We're focusing on head and neck cancer as the potential lead indication. Patients with locally advanced tumor, where we see that there is a huge unmet need. Simply, these patients, unfortunately, patients with locally advanced tumor in head and neck, they receive standard of care therapy, curative intent radiation therapy. However, they do not get cured over 40% of those patients, and the disease progress towards the metastatic stage with poor prognosis. So there is a great opportunity for TMX to modify hypoxia, overcome treatment resistance, and save thousands of lives. Again, I must say here that TMX is a pan-cancer solution and not limited to head and neck. Our second indication is GBM, which is very hypoxic, and we can actually pursue orphan drug designation. This technology is pretty much at the IND ready stage. We've done all the CMC work, repeated those GLP talks. We've met with FDA, got very positive response and feedback from the team. And now we are currently working on shaping the IND package for submission and initiating the phase 1 by early next year. And the phase 1 is designed to not only collect the safety data, determining the maximum tolerated dose, but also allowing us to collect the preliminary efficacy signal in combination with radiation therapy and the change in hypoxia by measuring MRI, which is a technique called MRI-IBIM. It will help us down the road to stratify the patients recruiting to phase 2 clinical trial. The second product in pipeline is a best-in-class agent, TERP-enabled trastuzumab. As you can see, we've conjugated TERP on the surface for targeted delivery, as well as encapsulated inside to improve the BBB penetration. And we have shown great preclinical efficacy, more than 50-fold higher drug penetration inside trastuzumab penetration inside the brain, significant penetration in the CNS area, in the lesions, and improving the efficacy of treatment by 6-fold and even achieving complete tumor eradication in some cases. This agent not only has multi-billion-dollar market opportunity, but also it creates a unique business development opportunity for us to pursue collaboration with biotech companies and improve other antibody penetrations inside the brain using TERP system. As I said, our strategies at CureCan is to create value and position the company for multiple value inflection points. And for that, we're raising 15 million Series A that supports the phase 1 clinical trial of TMX, collecting all those valuable clinical data set around the platform and the lead asset, IND enabling studies of the second product in pipeline, and also optimization of our TERP RNA leads, through which we can expect out licensing and early revenue generation through those deals. And as I said, we're gonna really prepare the company for multiple exit opportunities, from out licensing to collaboration, co-development with our strategic partners and IPO. I believe really that 3 things make a successful business: a great technology, a great business strategy, and a great team. We're so lucky to have amazing people at the company leadership team, people that have done this before, made unicorn businesses in the biotech field, and are helping us to make another success story. Thank you very much, and look forward to having your questions. And please do reach out to me if you want to discuss further.
Great presentation, Mohammed.
Thanks. Thank you.
We see I'm just looking at questions coming in from the chat, but before they come in, what, as a company, what is Kurkan looking for in potential collaborators? What is your ideal collaboration? What do you need to advance your platform?
Yeah, absolutely. I think the gene delivery side looks like a very interesting area for us at this point in time. It's one of our most active programs currently on the partnering side. We're actively looking for partners that we can develop, next generation gene therapies for targeting unmet, hard to treat diseases. So people, companies that have challenges with getting their payloads into the diseased area, I think we can help them out, and we can work together to make, big changes.
Right. You're in a very interesting area of, therapeutics, where, radiotherapy is really coming into its own right now. I just read something about Novartis's Pluvicto getting an FDA approval. So that is really fantastic. And, you're really in the right space right now.
Thank you.
So let me see if there are any other questions that have come in. I don't see any. If you would like to send your questions to Kurkan, please let me know. In the meantime, before we go on to our next company, I had some announcements to make. Thank you, Mohammad, for such a wonderful presentation. And
thank you very much. Thanks for having me today.
Thank you. Wonderful to have you. I want to announce that we are going to be launching our 2023 iteration of MassBio Drive, our wildly successful biotech accelerator. The spring applications open on January 3rd. If you're a scientific founder or you know one with a high potential therapeutic or platform technology, MassBio Drive can help you grow your biotech through BusinessNet, fundamental curriculum, expert training and mentorship, and ecosystem visibility over a quick but transformative 8 weeks hybrid program. All without any equity stake by us. It's all free and it's hybrid, so it's a global accelerator. If you're located in Massachusetts, welcome. If you're not, you're still very welcome. We'd love to have you. The MassBio innovation team is attending JPM, so if you're attending, let us know. We'd love to meet you there, and we'd love to connect with you. And we have a MassBio Innovation Week newsletter that goes out every week, every month. If you're not getting it, please go to our website, sign up for an email account. You don't have to be a member to do that. You can still get our email, and we'd love to see you at our events. With that, I'm done with my announcements, and I'd like to welcome our next founder, who's going to be coming in soon, Navneeta Navar from HDAX Therapeutics.
Hi, everyone. I'm Navneeta from HDAX Therapeutics. We are a preclinical stage company developing small molecule targeted therapeutics for neurological diseases with axonal dysfunction. And we hope to develop the first ever disease-modifying therapy for chemotherapy-induced peripheral neuropathy, which is our first indication. And this is the team leading the venture. I'm the CEO and co-founder of HDAX Therapeutics, and I have a PhD in medicinal chemistry and a first-time founder. That's where I built, during my PhDs, where I built a lot of the foundational science behind the company. And we've actually spun out of the University of Toronto last year from Professor Patrick Gunning's lab, quite a serial entrepreneur. And we are working with, so we've got a lot of experience in small molecule targeted therapeutics and HDAX6 intervention, and we're working with chemo-induced peripheral neuropathy, or CIPN in short, disease experts, both in the preclinical and clinical, who bring both preclinical and clinical expertise. So axonal transport dysfunction drives several neurological diseases such as ALS, peripheral neuropathies like chemotherapy-induced, diabetic-induced, inherited ones like Charcot-Marie tooth. And HDAC6 is a protein that's been broadly implicated in axonal transport dysfunction. Specifically for chemotherapy-induced peripheral neuropathy, it's a huge market, about 1 billion, just focusing on symptom management. And that's really because these patients do not have any other treatment apart from painkillers and physio. So at HDAC Therapeutics, we are targeting the protein HDAC6 to potentially become a first-in-class curative treatment for these patients and several others who face these neurological diseases that are driven by axonal transport dysfunction. So HDAC6 stands for histone deacetylase 6, but interestingly, it's actually not a histone deacetylase at all. It's a misnamed protein, which is more of a cytoplasmic lysine deacetylase and does not touch histone, so hence not an epigenetic modifier, and really quite its functions quite unrelated to the previous histone deacetylase proteins that have been targeted in the past. HDAC6 is actually implicated in some key pathological pathways that tend to drive neurological diseases. Firstly, it regulates transport in axons. It's important in cargo transport, both proteins and mitochondria through the axons. Secondly, it regulates mitochondrial dynamics and mitochondrial dysregulations. And very interestingly, it has a ubiquitin binding domain, which enables it to bind to misfolded proteins, and it behaves as a carrier protein for ubiquitinated misfolded proteins for autophagic protein degradation. So it essentially carries it to the lysosome when the UPS system is dysfunctional or overwhelmed. And these interesting roles in cells is what has made it a relevant target in chemotherapy-induced peripheral neuropathy and also the associated chemo brain or cognitive decline that we see in these patients. So full global knockout of HDAC6 has shown that in vivo, it can fully prevent cisplatin and other traditional chemo-induced neuropathy. What happens because of chemotherapy is that it leads to mitochondrial and axonal damage. That's mitochondrial dysfunction, axonal transport deficits, and that results in pain, so mechanical allodynia, and also nerve fiber loss. Intervention with HDAC6 inhibition or knockout could essentially stop this process, enabling us to possibly prevent this chemotherapy-induced peripheral neuropathy or even treat it. And although there's a couple of people who have tried and are trying to inhibit HDAC6, we bring in a very novel bitopic binding mechanism for HDAC6 inhibitor discovery. What I have here is a co-crystal structure of one of our older proof of concept molecules in purple in the pocket of the HDAC6 protein. It's in the catalytic pocket. And traditionally, all molecules existing molecules that are already either being used as pan-HDAC inhibitors or are being investigated for purely HDAC6 inhibition tend to bind through this one green arrow, so a singular interaction. What we've been able to do is design a second layer of interactions, as you can see from the second green arrow, and it's a direct enzyme inhibitor hydrogen bond first discovered and patented by us, and that enables us to really bind to the target in a bitopic fashion. So not just through the singular interaction, but have a second layer of interactions, and that's really enabled us to improve the drug-like parameters of these molecules in a great fashion, starting from improvement of action that's enabled us to design nervous system penetrating inhibitors, improvement of drug lifetime, so improvements in the pharmacokinetic properties to design much longer lasting inhibitors, and also expand the mechanism of action. Our most advanced assets in lead optimization are reversible inhibitors, but we've been able to explore this bitopic binding mechanism for HDAC6 PROTACs or degraders as well. We have the know-how to expand this bitopic binding mechanism to a platform for other targets. So existing molecules that are under investigation suffer from a lot of challenges, and they're mostly fall under three buckets, starting from efficacy because of for limited target engagement at your molecular level, the singular binding mode that everyone tends to have does not really allow you to engage with the target strongly or long enough, hence the limited efficacy. In terms of safety, selectivity for HDAC6 is actually very important in order to ensure that there aren't too many off-target effects. So the HDAC6 protein comes from a family of 11 proteins, and the rest of them are epigenetic modifiers. So ideally, you would just want to inhibit one while not touching the rest. And that's oddly difficult because of the huge structural homology between all these 11 proteins. And that is why previous molecules that have come tend to have a lot of adverse effects, because they end up inhibiting multiple proteins in this family of 11 proteins rather than HDAC6 exclusively. And finally, in terms of pharmacokinetic challenges, the existing molecules have a very hard time either crossing the blood-brain barrier, nerve-blood barrier, and surviving in the body for an extended period of time. And over the last 6-7 years of drug discovery in academia, and now after spinning out the company, we've been able to really differentiate across not just one, but in fact, all of these key challenges. Starting from efficacy, we've been able to design picomolar binders compared to the nanomolar binders that you currently see with leading competitors. In terms of safety, we've really prioritized true target selectivity, and we've been able to show that we have almost 800-fold HDAC6 selectivity over the whole family of 11 proteins. And in addition to that, we have also looked at other protein families such as kinases and hERG, where we've been able to show that we do not inhibit those proteins or interact with them at all. And finally, in terms of pharmacokinetics, that's really been a game changer because it's been awfully difficult in academia and industry to design blood-brain barrier or nerve-blood barrier permeable inhibitors. With HDAC6 potency and selectivity. And in vivo, we've been able to show half-lives of about 2 to 4 hours, so significantly higher than that of existing molecules with added BBB penetrance, so really opening up the avenue for neurological diseases. And here's some numerical values to show that how our potency, selectivity, so even at this preclinical stage, how our assets are truly differentiated from the inhibitors that are currently in clinical trials right now. As you can see, the IC50 of HDAC6 is how strongly we inhibit the target, and we've been able to show in the inhibition in picomolar range while in nanomolar range. As I mentioned, the selectivity for HDAC6 is almost 800-fold, so significantly higher than that of existing molecules. And we've also been able to design blood-brain barrier permeable inhibitors, and I'll get to the data in a moment. So in terms of target engagement and selectivity, both in vitro, as I mentioned, picomolar range inhibition with very high selectivity, and it has translated well in neurological cells in vitro. So acetylation of alpha-tubulin, this is a clinically validated biomarker that shows we use to show HDAC6 activity. So in this Western blot, you'd ideally want to see an increase in acetylation of alpha-tubulin to show on-target activity. What you can see here on the right with our molecules, even at 50 nanomolar, we show considerably strong on-target activity. But when we compare it with one of the most advanced existing molecules, it even at 1,000 nanomolar, its on-target activity in neuronal cells is not as strong as our activity at 50 nanomolar. For off-target activity, the clinically validated biomarker that's used is acetylated histone H3. As I mentioned, HDAC6 is a non-histone deacetylase, so increase in acetylation of histone H3 indicates that you are targeting off-target proteins. And ideally, you wouldn't want an increase in the acetylation of this off target. And that's exactly what we see with our molecule, even with a dose increase, that we don't really show a significant increase in acetylation of the off target. But when you compare it with existing molecules, and here's just one example, most of them tend to have an increase in the acetylation of the off target. Additionally, we also look at in-cellular residence time via nanobretta assay. So in live cells with HDAC6, the in-cellular residence time in HDAC6 for our molecules has actually been at least tenfold greater than most existing molecules, and we believe that the bitopic binding mode might have a role to play in the sustained duration of action that we are seeing in cells. In vitro, we've also looked at preliminary talks such as cardiac safety. We've been able to show that for our top molecules, we do not inhibit hERG at all. And in terms of genotoxicity, we've also been able to show an Ames a negative test. So when we originally started the work, we were mostly focused on oncology because that's where, yes, older pan-HDAC inhibitors really are still used and are clinically validated. But we quickly, as we studied HDAC6 inhibition for oncology in vivo, this is specifically an MM1, a xenograft study, so a multiple myeloma study, we quickly realized that with very selective and highly potent HDAC6 selective inhibitors, you're not likely to be an anti-cancer target. So our tumor growth inhibition wasn't the greatest, and that makes sense because HDAC6 is not really a protein that's whose inhibition will lead to a cytotoxic effect or high tumor growth inhibition. So those studies, both as single agent and in combination with bortezomib, although did not show true anti-cancer effect, I guess what we now believe that is correct, but we've been able to show on-target pharmacodynamics. So with an increase in acetylation of alpha-tubulin in vivo, with no change in acetylation of histone H3, so the two biomarkers that I introduced in the last slide. So we could show on-target activity in vivo, we could show there's no off-target activity in vivo, and on top of that, we could also show no toxicity in both the single agent and in combination. So this was really, although this wasn't a model where we could show disease-modifying therapy because we pivoted to indications where HDAC6 is a more relevant target, this in vivo study and actually another one in melanoma really showed us on-target pharmacodynamics and no concerns with toxicity with some of our best molecules. And in terms of in vivo pharmacokinetic profile, we've been able to show that in plasma, in vivo, as you can see on the left, in blue are our top two molecules, and in yellow is one of the existing molecules, a competitor molecule. We've been able to show that our half-life in vivo and plasma is actually about six-fold or even higher, like 6 times higher than that of the existing molecule, where, yeah, you're looking at between 2 to 4 hours in vivo, while other molecules are just about 15 to 20 minutes. And in addition to that, in the brain, in mice in vivo, we've also been able to show blood-brain barrier penetrance with a Cmax of about 4,000 nanogram per mil with an IP injection of 20 mg per kg. And the half-life is a little less than around an hour, and that's what we are trying to optimize right now. Existing molecules actually have almost zero brain permeability. And we've looked at our solubility, stability, permeability, and so on. And we've been able to show that with our top molecule, we actually we're not an efflux substrate. So our efflux ratio is 1, meaning that our permeability on both ends is very high, and we don't actively get kicked out by the blood-brain barrier. So, as I mentioned, our initial focus is on chemotherapy-induced peripheral neuropathy for market entry. We see the opportunity to be the first-in-class preventative and/or treatment, and we've picked chemotherapy-induced peripheral neuropathy out of really all the neuropathies, and I guess overall from the neurological indications that are driven by axonal transport dysfunction, because this has shorter clinical endpoints. So we do see the ability to show proof of concept in humans faster, and there has been a lot of this biological validation of HDAC6 in the preclinical stages specifically for this disease. So we do see that this could really make a difference, and it would be the quickest market entry. But following that, we aim to tap into bigger markets, but like diabetic-induced peripheral neuropathy, which is a $4 billion market and constantly growing, and also the inherited peripheral neuropathies like Charcot-Marie-Tooth disease, where it's a similar common pathological mechanism of HDAC6 playing a role in axonal transport and mitochondrial movement and deficit. So we do see that the same pathological mechanism plays a role in most of these neuropathies, and there's also the opportunity to be the first-in-class disease-modifying treatment for those markets, because they similarly suffer from not really having anything apart from symptom management. In terms of growth, we aim to expand towards other neurodegenerative diseases after, and interestingly, HDAC6 is a newly hot target in heart failure and cardiovascular indications, and that's an avenue we are looking to explore in the long run, and expand our pipeline to our other assets. So explore the biotopic binding mechanism for other targets, and look into HDAC6 degraders and covalent inhibitors as well. And here's the timeline for value creation. We've started the science quite a while ago, and only when it reached its inflection point and we could show true differentiation from what's out there, that's when we formed the company last year. And in the past year, we've been able to get the option to exclusively license our IP out of the University of Toronto, file our second patent with Wilson Sonsini to protect our nerve-blood barrier permeable molecules, and also have a pre-seed of that's enabling us to continue the lead up and do in vivo studies to show disease-modifying effect in chemo-induced peripheral neuropathy. Around spring of next year, we aim to have a seed round, and that should enable us to nominate our clinical candidate for our first indication, and also expand the pipeline to ultimately be the HDAC6 company for a range of diseases where HDAC6 has been an interesting driver, and its inhibition could really have some meaningful disease modifying effect and change the course of disease in several diseases. And yeah, following that, IND-enabling studies, and we're looking at a clinical trial in about 2025. So at HDAX, we're trying to develop the first ever disease-modifying therapy for neurological diseases with axonal transport dysfunction. I'm happy to take questions.
Hi there. Wonderful presentation again.
Thank you.
I'm just waiting for questions to roll in, but before they do, can you tell me what does the future look like for HDAX? What is your ideal collaboration? What do you need from investors or collaborators to take you over the next hurdle, your next challenge?
Yeah, well, that's a good question. And I think for HDAX Therapeutics, we named it HDAX because we ultimately wanted to be the HDAX6 company. Well, I guess initially the HDAX6 company, and maybe expand to other HDAC targets and non-HDAC targets as well. But we do see the value of this target in a range of diseases, and we see potential partners seeing that as well in neurodegeneration, in cardiovascular diseases, and more. So our goal is to drive this pipeline of brain permeable inhibitors, peripheral acting inhibitors for the peripheral focused indications, and partner with, interested partners in order to advance multiple programs at the same time for multiple diseases, because we do have different assets that target in different fashions in different parts of the body, so they would be ideal either for central acting molecules or peripheral acting molecules.
Yeah. So what has been your biggest challenge so far? in either raising investor, seed funding, or kind of presenting a platform to potential investors? And how do you think that you want to go forward?
I think one of the biggest challenges, in reality, I think challenges change as a startup from week to week. But I guess an overarching challenge that I think I would still consider a challenge is really on the indication selection. As a startup, it's definitely not a good idea to avenue into multiple different indications at the same time, even though the target's interesting and different potential partners want you in different avenues. But I think we're really trying to keep our focus while ensuring that we do have an open mind in order to, look into with our different types of assets that would be catered towards different diseases. We do have that in the pipeline, but as a company, we are trying to advance it towards an indication where we think we could bring the most value in the quickest fashion.
Thank you. Thank you, Navanitha. Thank you for joining us all the way from Japan today.
No problem. Apologies for the technical hurdle. I don't know, after so many years, it's still a problem.
I think, when you're doing a virtual live event, technical difficulties do happen, but I'm glad we could have you, and thank you again for joining.
Thank you so much. It was really nice. Thanks a lot.
I also want to thank everybody from our ecosystem that has joined us for our first day of our inaugural Innovation Week. We have a great program for the rest of the week. And we hope you'll also join our forum. That's on December 13th at 4:30. It's a year in review. We have some great speakers. We have Wade Brown from BMS. We have Omar Khalil from Sante Ventures. And we have a couple of other great speakers, including our very own Ben Bradford. So hope you'll join us, and we hope to also see you at the holiday party this Thursday. Thank you very much, and thank you for your patience as we navigated technical issues on the platform.